Hello and welcome to the session on neurogenetics. I'm Ammar Al-Chalabi. I'm a neurologist and one of the editors of Brain. We have many excellent speakers lined up for you today. It's a 1 hour 20 session. If you have a question for the presenter, please add it to the question box during the session and we'll try and cover as many as possible during the presentation. And if you want to chat with your fellow delegates, please don't forget you can use the chat box for that as well. But questions in the chat box won't be picked up for the Q&A session, so please remember to put them specifically in the question section. Our first presenter is Professor Julie Williams, CBE, from the University of Cardiff. She'll be well known to very many of you, if not all of you. She's a world leader in Alzheimer's disease research. She served on the MRC Neuroscience and Mental Health Board and the Scientific Advisory Board of the Alzheimer's Research UK. She was the Chief Scientific Advisor to the Welsh Government. She's the Director of the UK Dementia Research Institute in Cardiff. She was Dean of Research at the School of Medicine in Cardiff University, and a really amazing thing, I think Time magazine rated 2 of her research papers from 2005 and 2009 as within the top 10 breakthroughs of those years. She's leader of the IGAP Genetics Consortium, which has driven AD genetics research worldwide. So we're very honoured to have you speak with us, Julie. This is the teaching talk, and it's about what can we learn from genetics in neurology. Thank you.
I think it's a really interesting time to talk about what we can learn from genetics in the field of neurology and specifically in the field of Alzheimer's disease. So what I'm going to talk about is finding genes for Alzheimer's disease and how this is really informing our understanding of the disease. And I'm sort of capturing that in what I call a multiplex model. Then I'm going to talk about how we can use some of these findings in a variety of ways, including capturing the polygenic risk. That's the risk conferred by all the genetic evidence that we've so far accrued and how we can use that. And then to look specifically how we can go from the genes we've discovered to try and understand disease mechanisms. So just a little bit of background. These are very, very early findings now. The heritability of late onset Alzheimer's disease is between 58 and 79%, depending on what measures you use. And what's interesting is early onset is highly heritable with heritability in excess of 90%. These are some of the mutations, so it's displayed on our chromosomes. So you have APP was one of the first mutations identified, and this gene, along with presenilins 1 and 2, have shown a number of mutations, and I've given you estimates on the side. There's probably more mutations that have been discovered in some of these genes, but these are very strong genes, very strong effects. If you get one of these mutations, you will have about 99% chance of developing Alzheimer's disease, and you can almost anticipate within about a few years when the disease will begin. ApoE is another risk variant, a very strong risk variant that confers susceptibility to Alzheimer's disease. So this is what the picture was when actually I started doing my research. Genome-wide association studies have been really informative for more common forms of Alzheimer's disease, trying to find the whole picture of the genetic underpinnings of this complex disease. And just to give you an idea, what we do is we take a small sample of blood usually and extract DNA from that, and this gives you an idea of a variant on chromosome 1, and we have 2 copies of each chromosome, so we have 2 copies of this variant, and what we simply do is look at a particular SNP, a particular variant, and how frequent that variant is in people with Alzheimer's disease compared to what we see in a comparison group of people who are well. And this is to represent the population. And as you can see here, we have an association There are more of the red variants, if you like, that are associated with Alzheimer's disease than in the population. And if this was found in a good proportion of the sample that you use, then you can say you have an association. So you know that within a certain distance of that particular variant, there is a variant that is conferring susceptibility, that is affecting probably the disease mechanisms underlying Alzheimer's disease. But because we're doing this millions of times in each individual person's genome, we are making multiple tests. We need very powerful samples. So we have had to have extremely large experiments involving tens of thousands of Alzheimer's cases and comparison individuals to be able to show compelling evidence that we have genuine associations. Now, these are some of them that I've been involved in, and as you can see, the numbers grew as we were able to collaborate. And this is a testament to the vast amount of collaboration that has gone on across the world in the field of genetics. And that has enabled us to find a number of genes and really get a grip on what may be going wrong in Alzheimer's disease. This gives you this is a cartoon of some of these risk factors, susceptibility factors, and to date, we have 75 genome-wide significant loci or that we know are associated with Alzheimer's disease. That's a significant achievement, I think, in this area. This does not capture all of these, but some are yet to be published this year. So what does this mean? Now, these associations were not occurring at random, that there was a pattern here, or patterns developing within the data. And what we did some time ago is try to match the genes that were implicated by the associated variants and see if we were getting more hits in functional pathways than you would expect by chance. And indeed, we did show that. And one of the things that came out most strongly is the influence of the immune system. And that it seemed to be every time we found new genes, we would find immune relevant genes in the collection. Endocytosis, cholesterol transporter, other things that came up as pathways. And as time was going on, we did find some evidence that indirectly implicated the effects of APP, amyloid, and other things. But the main things that were coming out initially were immune relevant pathways. So we tried to capture this in some sort of model. This is just a summary of what we think the genetics at a gross level are telling us. And we propose this as a multiplex model of Alzheimer's disease. And as you can see, you have different components that are working together. So it's a combination of risk factors. It's not just one or two things going wrong. It's a number of things that are combining to tip you into a disease process. Immunity, as I've mentioned, is implicated. Endocytosis, cholesterol metabolism, protein ubiquitination, and A beta and tau also fall into this. Although we don't have genetic evidence for a vascular involvement, environmental studies really focus, I think, on vascular health. So I've put that in to give you a more complete picture. And some of the tissues that were coming out were interesting. When you compared the expression of the risk factors that we were identifying, we got a lot of them were being expressed in tissues like microglia. And that's something that we have now are continuing to follow up. There are other tissues that are involved in the disease. So pulling this together, in terms of the risk factors for Alzheimer's disease, you can see on the left here that around about 35% is estimated to be environmental, and the remainder, and it, as I say, between 58 and 79%, estimated to be genetic. So there's a significant genetic contribution. The familial AD loci, as I say, account for much less than 1% of all cases. But when you chop up the heritability, many of the genome-wide associated genes account for probably under 20% of the heritability. There's a lot of heritability in the category here, all other assessed SNPs. And that's something that we wanted to exploit, as well as looking at what the single genes or the single loci might be able to tell us. What about the whole gamut of genetic heritability? What can we capture that? And that's something I want to talk about now. So we attempted to capture this in what we call a polygenic risk score. And this is one of our original studies with Valentina Escott-Price played a significant role in this, and we looked at our sample of about 17,000 Alzheimer's cases and 37,000 controls, and created an algorithm that tried to capture all the genetic variants. So we went down to including anything that had an association with a p-value of 0.5. So below that, we weren't adding anything into the algorithm. So we ended up with about 87,000 SNPs in this algorithm. And when you put this algorithm together, we found that we could predict pretty well, and this area under the curve here. So if you added in all of the genome-wide association SNPs and the gamut of all associated SNPs, we could predict between 75 and 78% of the time. So that was correct at that level. Now, we wanted to see what would happen in other populations. So the GERAID sample is the sample I've described. That's the one I lead. And ADNI is the sample I think most people are familiar with. It's a very commonly used American cohort. And we also pulled together a pathologically confirmed cohort. So I'm just going to talk about that one because I think that's the most convincing. So if you just use ApoE on its own to predict whether you're going to get Alzheimer's disease, you would get it right just under 70% of the time. If you use the PUL polygenic risk model, you'd get it right about 80% of the time. And interestingly, if you then just chose people that are at the polygenic extremes, those with all the risk factors and those with the fewest risk factors, you could get it right over 90% of the time. Now that can be very useful for us in identifying people who are at high risk of developing the disease. And I'll talk a little bit about that in a moment. So what we decided to do was look at those at the extremes whose sensitivity is over 90% for Alzheimer's disease. So we looked at those that were 2 standard deviations above and below, and we standardized this on a population. We used the 1,000 genomes here, but you can use others. Okay. So one of the ways we use this is to create a resource of pluripotent stem cells that can be used by researchers. We call this the Ammar program. and it's part of the UK Dementia Research Institute. So again, we take a sample of blood and from cells we identify, we can change them into pluripotent stem cells into various others. For us, we're going to change these into microglia. But what we do is we take samples from individuals that have high polygenic risk. So these have got a heavy loading of genetic risk. And we also take samples from people at the lower end of polygenic risk who have very few of the genetic risk factors for Alzheimer's disease. We hope to create a cell bank of about 100 people with high and low polygenic score. And also we will include about 40 people that have very early onset Alzheimer's disease because they have a very strong genetic loading. So this is a resource that we hope will model the more common complex form of Alzheimer's, where we're looking at the combination of these different risk components, if you like, and how these work together to result in disease. So this gives us a little summary, this slide, of what we may do taking some of the genetic findings forward, looking at individual genetic variants, mutations or coding variants. And I must highlight the complication here in that many of the genome-wide significant variants that we've identified may not be obviously functional. They may be just signposting that particular region. And within that, there will be one or two or more functional variants that confer susceptibility to Alzheimer's disease, but we may not be able to identify what is functional from the genetic association. So that's been a challenge. And probably that's why having the combined polygenic score from individuals with high polygenic risk sort of overcomes that to some extent, because you're looking at all the relevant associated variants without having to identify which they are specifically. Okay, so coming back to the individual genetic variants, we can create cellular models, and indeed we have on not only from the mutations that have been known for some 30 years, but functional or coding variants like PLCG2. There was variant in, well, there's a couple now in that gene that we knew protected against Alzheimer's disease. There are others in ABI3 that increased risk of developing Alzheimer's disease. And we have modeled these and are modeling them in cellular models and animal models. Now they're learning some interesting potential mechanisms. It's too early to say anything definitive, but it appears that the sort of balance between endocytosis and phagocytosis. Phagocytosis is more aggressive form of immune response, that those at risk of developing Alzheimer's disease may have more of a balance towards phagocytosis than endocytosis. But that's early days yet. But these things are coming out of the very specific cellular modeling and animal modeling around single variant, single genetic risk factors. As we just touched on, looking at multiplex or a number of risk factors together, and this really better reflects what happens in common Alzheimer's disease. It's multiple things working together. So we can model all of it with the high, just set the high and low, but we could also in the future look at these different pathways and look at individuals that have a really strong loading on some, but not other pathways. But that's for the future. And we can take this information into high-risk epidemiology. So you can now, by looking at those with very high polygenic scores, identify individuals in their 20s, 30s, 40s, 50s in the early stages of the disease. And we know the Alzheimer's disease starts 15, 20 years before you see any symptoms, memory symptoms, etc. So to help us understand the early stages of disease, but also identify people for future clinical trials, because that's where you want to attack the diseases before it has done a lot of damage. So I think the genetics is opening that door most decidedly for common Alzheimer's disease. And of course, you can drug testing, looking at the pluripotent stem cells. You could envisage testing these and seeing if you can rescue some of the mechanisms or phenotypes that are observed in those at high risk of developing the disease. And that's another function that comes out of our current genetic understanding. And again, you can try and understand the mechanisms through cellular models, chimeras, putting these cellular models into animals, co-cultures, organoids, etc. But also, I think it's important specifically because we are implicating a lot of immune relevant mechanisms to take this back into live brain tissue, because immune systems, microglia, etc., are affected by their specific environment. So we need to do that at some stage in our modeling. So there's a lot of work to do, but some exciting paths to follow, I think, resulting from the genetics that we hope will help us understand disease mechanisms and produce new therapies in the future. So to summarize, genetics can provide a crucial link between biology, biomarkers, and human disease. Over 75 susceptibility genes have been discovered for Alzheimer's disease to date. Genome-wide association studies capture about 50% of the heritability for AD, so it's a good chunk of the variants. Polygenic risk score predicts Alzheimer's disease with around 80% accuracy in the general population, but can show over 90% sensitivity at the polygenic extremes. New iPSC models of common Alzheimer's disease based on high and low genetic risk will be becoming available shortly for researchers to really try and understand common Alzheimer's disease. And we can use this strategy to identify people at the highest risk in the earliest stages of disease development. Immunity or inflammation are specifically implicated, and the role of microglia is strongly implicated by the genetics. So we've pulled these together in a multiplex model to really hammer home that it's a number of components that need to be deficient, if you like, to develop the disease. Our understanding of disease mechanisms will be informed by exploring the effects of single genes and combination genes in the future. So I think things are looking quite exciting, and I'm thankful for these are just some of the people that have been involved in some of these studies, and some of the people that have supported and funded our work. And thank you very much for listening.
Julie, thank you very much for an absolutely wonderful talk. It was fascinating. We've got a number of questions, and I wonder if I could put them to you. So the first one is, you mentioned that the immune system and cholesterol have been implicated in the pathways that you've discovered in Alzheimer's disease genetics research. And I know those same pathways have been implicated in other neurodegenerative diseases like ALS and Parkinson's disease. Do you think that signifies a common underlying mechanism for all neurodegeneration? And if so, how can you explain the fact that they're cell specific if they've got the same kind of underlying causes?
I think we're talking about the same pathways. We may not be talking about the same individual combination of mechanisms, because we're not talking about one mechanism here. There are a number of things we know in Alzheimer's that may be changing, may be different in those that are susceptible. I don't think it's going to be the same. but I think it's implicating this area in general, contributing. And what we've also found, Caleb Webber in my group has shown that the risk factors for Alzheimer's disease, and these are mainly the immune ones, affect your trajectory in Parkinson's disease, but they don't affect whether you're going to get Parkinson's disease or not. Yeah. So there are complicated relationships, but it's just saying, this is where we need to be looking.
Okay, so you mentioned a kind of split between risk and progression, and I think it is very important. Do you find separate genes for progression in Alzheimer's, and how would you do that?
We're looking at progression. Progression is a little more difficult because we can't quite get the same amount of power. We don't have the data sets with longitudinal data. Not the same number that we have that are cases and controls, but it's something that we are looking into. So I can't give you anything definitive there in terms of whether they will be the same, but certainly that there is a difference with Parkinson's disease. So I suspect it's going to be a slightly different story.
Okay, thank you. We have got a question from Zhongbo Chen, who has asked about diagnostic accuracy. So in some diseases, the diagnostic accuracy is extremely reliable, and in others, you can only really make a pathological diagnosis and you have to guess the diagnosis in life. And I think Alzheimer's probably fits into that second category. How does that affect the reliability of genome-wide association?
Well, we've looked at that when we started out the research, diagnoses that we were using. We tested those against neuropath confirmed samples, and we were accurate over 90% of the time. So there will be about 10%, but in part that's why we need very large samples, because you can deal with the noise there. But I would there are groups that have looked at proxy diagnoses that using very powerful samples like biobank, etc., where the proxy diagnosis is whether your parent had some form of dementia. Now, I think we have to be wary about how we use those data. If we see relationships and associations that are also seen in cohorts that have used research diagnoses for Alzheimer's, then I think we can take those as supporting probably an Alzheimer's association. But there are many things that cause dementia, and there will be noise in those data. So we need to be careful about how we use those. But I think the level, the diagnoses that we are using for most of the GWAS studies are pretty accurate, but the power of the studies can mitigate any noise there.
Yes, you really do have huge numbers in Alzheimer's for your studies. So I was interested in your extremes polygenic risk score strategy. It's a very clever one. Do you think there's a role to make organoids? Because I noticed as well you were talking about neuronal and glial transformations, but mainly about glial cells. So presumably Alzheimer's disease requires both neurons and glia to have some interplay.
Yeah, it's a very good question. I think that's where we want to get to. I think we would look at two-dimensional work first with two different sorts of cell, because you need to get the precision. With organoids, you don't quite get the same precision in terms of your measures, but you do get a more realistic combination of cells. And what we want to look at is high risk in different tissues, in neurons, in glia, in astrocytes and microglia, and seeing what the combinations are with different environmental triggers as well. I think we need to look at what may be going on in the brain and try and mirror those as well.
Fascinating. Thank you very much, and thank you again for a really very, very interesting talk. Thank you. So now we move on to our next speaker, which is Dr. Avi Fellner, who's a consultant neurologist and medical geneticist at Rabin Medical Center in Israel, who's currently a visiting clinician scientist in Sydney. And he's going to be giving a data blitz. So it's a very short, sharp, shock type talk. He's going to be discussing in silico phenotype prediction by normal mode variant analysis in TUB4A related disease. So over to you, Avi.
Hello. I would like to thank for the opportunity to present this work in which we investigated the use of normal mode analysis in TAP4A related disease. There are no conflicts of interest to declare. TAP4A related disorders show a very wide phenotypic spectrum. While in one end of this spectrum there's an isolated adult onset dystonia, DID4 torsion dystonia, at the other end there's a severe infantile onset hypomyelinating leukodystrophy with associated encephalopathy. Between these two, there are cases of hypomyelination either with or without atrophy of the basal ganglia and cerebellum with variable clinical severity. This prominent phenotypic heterogeneity makes TAP4A a good candidate for the investigation of genotype-phenotype correlations. Previous works have suggested a potential genotype-phenotype correlation based on data from several variants. In this study, we performed a systematic NMA-based investigation of such correlations for all reported variants in this gene. Normal mode analysis, the method we use in this study, is based on the physical principle that every system or structure has a natural pattern of oscillating motion, and this is true also for the three-dimensional structure of proteins. It's a computational method that allows us to investigate protein dynamics by quantifying this motion pattern and the way changes in different amino acids in the protein affect this pattern, which in fact is their impact on the protein stability or its entropic profile. It's important to note that the use of this method is limited to missense variants. Through a combined search in PubMed, ClinVar, and NOMAD, we included in our analysis all reported pathogenic TAP4A variants with available associated clinical data, as well as the reported benign and likely benign variants in this gene. We divided the pathogenic variants into 6 phenotypic subgroups based on the type of the associated phenotype and its severity. Based on the NMA results of the wild-type protein structure and of each of the mutated structures, we obtained the NMA-based entropic profile of each variant. We then identified 11 partially overlapping structural clusters of variants based on their proximity in the 3D protein structure, and performed a pairwise cross-correlation analysis of the NMA results between all variant pairs in each of these structural clusters. We found a clear pattern correlating the genotype as expressed by the NMA results of each variant with the phenotypic subgroup, where across each of the structural clusters there was a continuum of this correlation, meaning that the higher the correlation of NMA results between two variants in the same structural cluster, the higher is the likelihood that these variants are associated with a similar phenotype. Except for 2 instances, this pattern was also kept in overlapping transition areas between structural clusters. Importantly, in addition to genotype-phenotype correlation, the NMA results clearly separated benign and pathogenic TAP4A variants. To summarize, we used NMA to integrate protein dynamics in the investigation of TAP4A related disease and demonstrated the applicability of our approach in the investigation of genotype-phenotype correlation, as well as its potential contribution to variant pathogenicity interpretation in this gene. In addition, our approach can be potentially utilized to investigate genotype-phenotype correlation and variant pathogenicity in additional monogenic conditions. To conclude, I would like to thank my dear colleagues who took part in this study and to you for listening, and I'll be happy to take questions. Thank you.
Thank you very much, Avi, for a really, again, interesting talk. So it's a fascinating method. You've basically taken protein modeling to the next level by modeling how these oscillations affect the clinical manifestation of that protein change. How feasible is it to do that proteome-wide? Is it a very computationally intensive task or something that could be done relatively easily?
We are trying to develop this as a more automatic approach. For now, it's probably not possible to do it proteome-wide, but it's something to think of course, but for the future.
And do you think that taking, for example, you've used TAP4A, which is implicated in the disease I study, ALS, if you take that and you say, well, it's causing this phenotype, does understanding how these clusters occur tell you about the mechanism of disease, or is it simply an observation that's kind of interesting? Can you use it to understand what the disease, why the phenotype happens, and therefore target a drug to it?
I think it depends on the specific group of diseases. For example, in epilepsy, where we have we can use this to characterize ion channels, we can get some more idea of the mechanism. But for this specific example that we chose, because of the great phenotypic heterogeneity that allowed us to do this research, it's not exactly telling us directly regarding the mechanism, but it's something again to get into in the future, that it's very initial.
I mean, I can see another use for it potentially, which is one of the systems that's developing, the ClinGen database, for example. There's curation of different phenotypes with their genotypes. And here you've got something that links genotype to phenotype, and quite often finding the evidence that a particular gene variation is relevant, particularly if that gene is pleiotropic, so that it can cause, for example, VCP mutation can cause multiple phenotypes, Paget's disease and a neurodegeneration type phenotype. Understanding why those are happening and what is linking the two can help with that kind of classification. I think it's a fascinating method. Thank you very much.
Thank you.
And thank you for giving us your talk. So we're now going to move on to the next speaker, who is Professor Janine Kirby from the University of Sheffield. She's a professor in neurogenetics, using gene expression profiles to understand neurodegeneration and to develop biomarkers of disease and progression. She's a world leader in ALS neurogenetics. I am privileged to have known her for many years and worked with her in that role. And she's going to be giving us the basic science research talk, or the discovery science research talk, on the use of microRNA in understanding neurological diseases. Over to you, Janine.
Good morning, everyone. I'd like to start by thanking the organisers for inviting me to give this talk on the use of microRNA in understanding neurological diseases. For the purposes of today's talk, I have no conflicts of interest. I'm going to start with a very brief introduction to microRNAs, focusing more on what makes them an ideal biomarker, as well as some of the considerations that should be taken when profiling their expression. I'll then go on to discuss some illustrative examples in amyotrophic lateral sclerosis, or motor neurone disease, to highlight how microRNA profiling is being used in neurological disease. And then I'll end with some future perspectives of their use. Mature microRNAs are generally 20 to 25 nucleotides in length and regulate gene expression by binding to the messenger RNA, repressing protein translation. MicroRNAs are not fully complementary to the gene, and most have a seed region for target recognition. This allows each microRNA to bind to multiple different targets and for each gene to be regulated by different microRNAs. MicroRNAs have been identified as useful potential biomarkers due to the fact that they are stable through freeze-thaw cycles, unlike messenger RNA. They are also found in liquid biosamples such as blood and its derivatives, serum and plasma, as well as CSF and urine, and can be detected by multiple different technologies, making them easily accessible to study. As we now find more microRNAs, this is accompanied by an increase in our understanding of their function. Unlike in other diseases, it is impossible to sample the brain in neurological disorders during life. We therefore rely on sampling liquids such as blood, CSF, or urine, as well as taking skin biopsies to generate induced pluripotent stem cells or induced neural progenitor cells to grow the neuronal and glial cells. However, as well as considering the source material, there is also whether you look at the whole solution or focus on exosomes, small membrane-bound extracellular vesicles that are secreted by the cells. When extracting RNA, specific kits should ideally be used in order to capture the small RNA molecules that are usually discarded when RNA purification columns are used. With regard to technologies that can be used, these include quantitative PCR, microarrays, and small RNA sequencing, and I've used all three. Each has their own advantages and limitations, But ultimately, it's the experimental question which will drive these decisions, as well as cost and the funding versus the funding available, and whether equipment is available in-house or requires outsourcing. So moving on to microRNA profiling in neurological disease, this is clearly a huge field for a 15-minute talk. However, a brief look at the literature shows multiple papers and reviews on the use of biosamples from Parkinson's and Alzheimer's disease, multiple sclerosis, and ALS, looking for both microRNAs and other non-coding RNAs as biomarkers to identify diseases, to identify those dysregulated within the disease. The number of papers is illustrated by this quick PubMed search, which showed there were nearly 3,000 papers available when using the search terms microRNA and neurodegenerative disease. And of these, there were 479 in the last year alone. As such, this means that now meta-analyses of papers are being published, such as this by Takousis and Sadlon, where 147 data sets from 107 papers were compared, and 52 microRNAs were consistently differentially expressed in samples of brain, CSF, and blood. A further 5 were differentially expressed in both the brain and blood. However, of those 47, 13 were not named in abstracts, and therefore actually going back to the original source papers is important when looking for support or not for your microRNA to be differentially expressed in your disease. Moving now to some illustrative examples where microRNAs have been profiled in ALS. The first two are work that has been carried out by my group in Sheffield, and my last one I wanted to comment on is Ido Margan's paper that has been recently published in Nature Neuroscience. Our study on microRNA profiling in serum was carried out to determine if we could identify a diagnostic marker for ALS. In addition to profiling sporadic ALS and neurologically normal controls, we also recruited 6 to 8 patients from a variety of ALS mimics, including the non-inflammatory neuropathies and myasthenia gravis. The others are listed in this down here. We determined the expression level of 750 microRNAs using the TACMAN low density arrays or tilde cards, which is a qPCR-based method. 12 microRNAs were found to be differentially expressed between controls in blue and ALS in yellow. We also found that there were microRNAs that were differentially expressed between the mimics, such as between the ALS, again in yellow, and the myopathy in turquoise in panel B, and between the ALS and the non-inflammatory neuropathies, such as those in green shown in panel C. 3 microRNAs were validated in a second cohort, and these were microRNA 206, 1433P, and 374B 5P. They were then assessed to determine if they were altered in response to riluzole, they weren't, or if they altered during disease progression. What we found was that microRNA 1433P in serum samples taken from sporadic patients later in their disease, this is at least 3 months later from the baseline, were increased, whereas the microRNA 374B 5P was decreased in samples taken later in disease. In contrast, however, to studies in the SOD1 mouse model, MIR206 did not reach a significance with an increase over the course of disease. We have also looked at changes in microRNA profiles of lymphoblastoid cell lines from patients with long and short survival, where short survival is defined as less than 1.5 years and long survival as over 6 years. Using small RNA sequencing, we wanted to determine which microRNAs were associated with the long and the short disease duration and which genes do these target, as this may identify an insight into the mechanisms of disease survival. And these may then subsequently provide prognostic biomarkers. So this work is unpublished, and we've recently reanalyzed the data and identified 26 microRNAs that were increased and 8 microRNAs that were decreased in patients with long survival compared to short survival, using a fold change of more than 1.2 and a p-value of less than 0.05. We then looked at which genes these acted on. And interestingly, there were pathways which are highlighted in grey, which were common to both the 26 microRNAs that were increased and the 8 microRNAs that were decreased. Just looking at these in a little more detail, taking the transcriptional misregulation in cancer as an example, 18 of the 26 microRNAs that were increased target a total of 59 genes, whilst 7 of the 8 decreased microRNAs target a further 51 genes. Further work is now underway to establish which these genes are. However, if you have a look at the KEGG pathway, and I appreciate this is very small, but what you can see is this is actually made up of multiple different pathways. Therefore, actually looking at which microRNAs act on which genes and whether they're positive or negative, regulating the pathways is important for understanding the function. The final example is, as I say, Ido Margan's recent neuroscience paper working with Owen Hornstein and collaborators, which highlighted the potential use of microRNA as a 181 as a prognostic biomarker for disease progression in ALS. Initially, sequencing was performed on plasma samples, which identified 125 microRNAs that were relatively stable during disease duration. A second study profiled a larger cohort of patients, which were divided into discovery and validation cohort. What they found was that low levels of microRNA were associated with longer survival, and high levels were associated with a shorter survival. This was replicated in the second cohort, and in the merged cohort you can see the graphs here. Interestingly, microRNA 181 is highly expressed in motor neurons, and by subsequently combining microRNA 181 with that of the neurofilament light, which is an upcoming biomarker in ALS, it was demonstrated that the combined data predicted a better survival than either biomarker alone, and better than 8 other clinical features that were previously shown to be informative. So just to end on future perspectives, so it's clear that there are some potentially prognostic markers coming through, and what we're also hopefully some diagnostic biomarkers that can be used clinically. In addition, microRNAs are now being considered as therapeutic agents, with microRNA mimics designed as duplexes which bind to the RISC complex and interact with the microRNA target, the messenger RNA target. The most common inhibitors are anti-MERS, which block the activity of the target microRNA through perfect match binding. Finally, microRNAs are being used to inhibit messenger RNA, such as Huntington, C9orf72, or SOD1, and these are currently in clinical trials with mixed results being reported. I'd just like to finish by thanking all the patients and the participants who have donated samples, members of the CITRAN who contributed to the work, and our funders. Thank you very much.
Janine, thank you very much for a really interesting talk, actually fascinating. And biomarkers are such an important and emerging field, and particularly microRNA, I think, is going to be a very strong contender as an excellent source of biomarkers. I wondered, you mentioned serum and CSF expression. I mean, you'd expect that there'd be quite a strong correlation between the two. Is that always the case?
Not necessarily. In the experiments that I've presented today with the serum, we've used different methods. So our serum work was using quantitative PCR and specifically only looking at 750, whereas the CSF work that we've done has been using small RNA sequencing. So there will always be some challenges in correlating that data. What is interesting, having whilst preparing this data, I also looked at our CSF small RNA sequencing, and we did find that supportive data for microRNA 181 from the Erin Hornstein's and paper, with that also being highly expressed in CSF too. And in our cohort, we did find it was increased, but not significantly. But we obviously had a much smaller cohort than the data that was presented by Erin. So certainly our data does tend to support that. And for anyone who's eagle-eyed, certainly there was a microRNA 181D, so a slightly different form, which was also reduced in expression in the long survivors compared to the short survivors. So again, there's clearly supporting evidence, but there are multiple different sort of isoforms of MIR181 as well. So I think it's a lot more work around that area that's going to be due.
So you've kind of touched on two important issues there that are related, which is to do with sample size. We've heard already the genetics talks today. We've got tens of thousands of samples for most complex diseases. But for RNA, it's more expensive to extract. It's more fussy about how you extract it. It's more fussy about storage, etc. So you've got that aspect, and you've got the fact most patients don't really want their spinal fluid tapped, and certainly controls are much more reluctant than patients. And so you've got, if you're looking at something that has to be collected from CSF, that can definitely be a challenge for RNA. But can you think of any ways that as a community we can kind of improve that so that we do get up to the tens of thousands? Because otherwise you will always have this problem of small sample sizes and therefore uncertainty.
Yeah, I would almost turn that round a little bit. So I presented the paper that they'd done this meta-analysis in Alzheimer's, and apparently they've also done a study, the same authors looking in Parkinson's, where they've been comparing the CSF with serum and blood as well. So I think ideally, if we can find microRNAs that act as biomarkers that are consistent. Now, it may be that you want to validate that in a smaller cohort of CSF, but actually, as you say, getting blood samples, and particularly from controls and the disease mimics, and ideally to have a biomarker that even as a sort of early flag to a general neurology clinic or even at a GP surgery to say, well, it might be something, and to reduce the delay, that diagnostic delay. If somebody is fast progressing, then actually it's a lot quicker than someone who's slow. So if it will never actually sort of replace a clinician in that diagnosis process, but maybe speeding it up and clarifying in a more general neurology clinic that people need to get to one of the specialist centres, I think is where it's going to help. And potentially also in clinical trials where there might be early readouts of whether particular drugs are having a positive effect rather than a negative in, so taking longitudinal sampling, I think, is key. So many previous biomarker studies have been just between even just disease and control, not even including mimics and other neurological controls. So I think where we're going with collecting longitudinal biomarkers and is the way to go.
Yeah.
I think coming back to your earlier comment, microRNAs, because of their stability, look to be more beneficial than, say, messenger RNA. But I think validation is always key in these studies.
Yeah, and I like your point about clinicians, because I think for some of these conditions, particularly neurodegeneration, it's a very clinical diagnosis. And having a biomarker that is better than a clinician, how would you ever prove that very easily? I think it'd be difficult. But having a biomarker that at least is sensitive enough that a GP or a primary care physician or a non-specialist could say, well, I've got a high index of suspicion, let's refer early. I think that's a very valuable role for diagnostic biomarkers. And for prognostic biomarkers, I always think of them as having multiple roles. So for example, you could use them to stratify clinical trials, you could use them to understand mechanism, which you touched on, and you could use them as endpoints. But I think for treatment response, which would be the ideal thing for speeding up clinical trials, don't you really need a biomarker that's generally stable through disease, or at least predictable? Do you think we have that for microRNAs?
Short answer is I don't know. I mean, in identifying the MIR181, they did try to find one that was generally initially not that variable around patients, but they then found it did increase over time. So I think you need to have something that is differentiable during the disease, and whether you take long versus short as an initial, or you know, I think there's more work that really needs to be done to establish that. Okay,
well, thank you very much, Janine. That was a fantastic talk, as expected. So thank you for your time today. So we're now, we would be going on to Shan Wang, but unfortunately they're unable to make the presentation. So we're going to jump straight over to Professor Deb Powell's talk. So he is a pediatric neurologist at King College London and a colleague, and he's a clinician scientist researching the genetics of common epilepsies with complex inheritance. He's driven the field really, and has made several interesting discoveries, which you'll hear about shortly. He's going to be giving us the clinical translational research talk on stratified medicine research in epilepsy. So over to you, Deb.
Welcome to the 2022 Brain Conference. My name is Deb Powell, and I'm going to be speaking today about stratified medicine in epilepsy. I'm a paid consultant to UCB Pharma and Engrail Therapeutics. What is stratified medicine? The mission of stratified medicine is to identify groups who will benefit most or be harmed least by specific interventions. The challenge in clinical medicine is in deciding on the management of the individual case when the evidence that we have before us refers to the average. Prognosis and prediction of treatment response are particularly important in epilepsy because it's a common disorder. There are 600,000 people with epilepsy in the UK. Epilepsy is classified into roughly 30 different epilepsy syndromes, and prognosis varies not just between the syndromes, but also between individuals with the same syndrome. The status quo with anti-seizure medicines is not very good. Of patients who are prescribed their first anti-seizure medicine at diagnosis, only about 1 in 5 find this first prescription to be both effective and tolerated. About 40% of patients experience adverse effects that leads to withdrawal of the drug. And this trial and error approach leads to considerable patient harm, as well as wasted resources and time. Stratifiers are variables that differentiate risk of outcomes. They may be found in the patient record, for example, demographic and clinical variables like sex or age of onset. They may be not immediately obvious, endophenotypes, and in epilepsy we have the EEG and neuroimaging. They may be molecular variables like HER2 receptor status in breast cancer, lipids for cardiovascular disease, SCN1A variants in epilepsy. They may be environmental, such as exposure to ultraviolet light or diet, or they may be derived from computational or artificial intelligence methods, such as mining of big data sets or meta-analyses of primary research. I'm going to be talking about juvenile myoclonic epilepsy, which is a common epilepsy arguably a women's disease. It has its onset in adolescence and persists well into middle age. Some characteristic features of JME, as it's known, are that many patients report their seizures are triggered. In other words, they don't occur spontaneously. About 1/3 experience resistance to anti-seizure medicines. And side effects are common to two of the most commonly prescribed drugs, levetiracetam and valproate. And last, because of its onset in adolescence and the involvement of prefrontal circuits, impulsivity is a feature of JME, which can lead to several different adverse psychosocial outcomes. So I'm going to present two studies from our work. One asking the question whether seizure triggers are associated with resistance to anti-seizure medicines. And the second study asking whether impulsivity is associated with psychiatric side effects to the drug levetiracetam. So the first question, stress-sensitive seizures as predictors in JME. And for this, we draw upon the BioGene study, which is the largest clinical genetic study of JME, with more than 900 participants across 75 sites in 15 countries. As we know from the epidemiology of JME, about 2/3 of participants were female. And if we look at lifetime exposure to anti-seizure medicines, we see a clear sex difference. So, for example, valproate is much less prescribed in women than in men because of risks of heterogeneity, whereas levetiracetam and lamotrigine are both prescribed more in women than in men. And topiramate is a small player. If we look at drug resistance, we see there are similar sex differences between women and men, with more drug resistance, about 35% compared to 30% in men. And if we look at seizure precipitants, that is environmental triggers, as well as laboratory provocation, we see that the distribution of these precipitants is largely overlapping in individual patients. So, for example, sleep disturbance, menstrual cycle, alcohol consumption, stress, mental concentration, visual or photic sensitivity, and the photoparoxysmal response in the EEG laboratory. And when we look across all of these precipitants, we see, again, sex differences. Women more likely to have seizures in response to laboratory provocation, more likely to have photoparoxysmal response in response in the EEG laboratory, and taking all triggers together, a higher frequency in women compared with men. And if we break down the triggers into the individual items, we see that sleep disturbance and alcohol consumption are in equal frequency for the sexes. But for women, stress, photosensitivity, and obviously menstrual cycle are more commonly reported triggers in women than in men. If we now make a multivariate model of drug resistance, we see that amongst men, absence seizures is the strongest and only independent risk factor for drug resistance. And this is a well-known factor in the idiopathic generalized epilepsies. If we look at women, the pattern is slightly different. Absence seizures are there, of course, but stronger risk factors include menstruation, mental concentration, generalized tonic-clonic seizures, and stress. If we now further stratify both on sex and on the presence of absence seizures, some very interesting patterns emerge. Here on the left, we look at females with absence seizures, no other factors are significantly associated. But in women who don't have absence seizures, then mental concentration and menstruation emerge as very strong associations with odds ratios 35 and 15. Stress and sleep deprivation remain as very strong triggers, very strong associations in women without absence seizures. And this leads us then to a predictive model of drug resistance in JME, where overall a third of participants are drug resistant, with some small differences between the sexes. Absence seizures doubles the risk of drug resistance, both in men and in women. But then stress doubles risk of drug resistance in women only who don't have absence seizures. And then photosensitivity halves the risk of drug resistance only in those people who have either absence seizures or stress-related seizures, but not both. So in summary, stress triggers may predict drug resistance in JME, but there is a hierarchy of risk in that stress modifies risk only in women who don't have absences. These findings are congruent with evidence from other research, which shows that stress chronifies migraine in women selectively. And we know from neurobiology research over the last 2 decades that women's stress response is qualitatively different to that in men. We have some limitations in this study. It's cross-sectional, so we would need prospective validation. And triggers are, of course, subjective phenomenon. The questions these findings raise, though, are whether stress management would reduce risk in this subgroup of women who don't have absences but do have stress-sensitive seizures, and whether we should be managing women's epilepsy in a completely different manner to that in men. Let's turn now to stratifiers of adverse drug effects, specifically impulsivity. Returning to the BioGene study with its sex distribution, and these, again, as we showed before, the drugs that are commonly used. This graph now looks at the adverse effects self-reported, and we see, again, sex differences in self-reported weight gain attributed to valproate, almost double in women than in men. Psychiatric adverse events to levetiracetam, more common in men than in women. Rash, same in both sexes. A disproportionate amount of adverse effects to topiramate compared with the overall proportion of prescriptions. And if we look at valproate weight gain in a little bit more detail, on the right here we see the WHO BMI categories, and we see in the pink those patients who have not been exposed to valproate, who've been exposed to valproate but don't report weight gain, and in the red, those who self-report weight gain with valproate. And what we see is that those who are who report weight gain with valproate are more likely to be in the obese category. And remember that every shift from normal to overweight and obese carries a serious consequence for risk of all-cause mortality. If we now look at levetiracetam, it's one of the top 100 prescribed drugs in the world. And in the US alone, there are 1 million patients taking levetiracetam at any one time. In generalized epilepsy, levetiracetam is easily the most prescribed drug. And looking at the psychiatric adverse events that are associated with its use, irritability, anger, aggression, depression, personality change. And these are more commonly seen in youth and also patients with a previous history of psychiatric disorder. So we're looking at impulsivity to see if it's associated with these psychiatric adverse events. And just as it sounds, impulsivity is the tendency to act or make decisions on a whim without regard to consequences. The neurobiological model involves several nodes in the limbic network, but predominantly a top-down inhibition from prefrontal cortex to both the ventral and dorsal striatum. Impulsivity is a moderately heritable trait, and in the general population, impulsivity is associated with difficulties in psychosocial outcomes, including in relationships, career, education, and increasing the risk for risk-taking behaviors. It's a key feature of several neuropsychiatric disorders, including ADHD, bipolar and personality disorders, and addiction. We measured impulsivity through a very well-validated self-rating scale known as the Barrett Impulsivity Scale. 8 items scored up to a maximum of 24. And firstly, we found that impulsivity correlates with seizure frequency. So the difference between daily seizures and no seizures is a difference of 4 points on the Barrett scale. Comparing with other neuropsychiatric disorders, we found that patients with JME have an impulsivity score around the same as that found in patients with neurotic or personality disorders, but lower than that seen in ADHD or other psychoses. In multivariate analysis, we found that after adjusting for seizure frequency and gender, a BIS score with a cutoff of 21 was strongly associated with odds of an adverse psychiatric event, levetiracetam, with an odds ratio of 2.3. However, its properties as a predictor are less good. The BIS score of 21 gives us only a positive predictive value of 50%, which is not really usable for clinical translation. So in summary, impulsivity is a poor predictor of levetiracetam side effects, although it is strongly associated with these in both men and in women. Our findings replicate a previous study which found a similar association in a similar population. Impulsivity might be a proxy for other psychiatric diagnoses which have impulsivity as a core component. And we're left with the questions in the future of whether a genetic susceptibility marker would perform better as a predictor, and whether we should be considering a multifactorial model of drug side effects. So in summary, when it comes to predictors of drug resistance, sex has definitely been overlooked as a stratifier. Stress may be an important stratifier and may be an important treatment target. Sleep disturbance and the menstrual cycle are well-known risk factors, perhaps not so well-known as disease modifiers, but we don't have currently evidence-based treatments that interventions against these factors would modify disease. When it comes to adverse effects, we have very few biomarkers to predict the incidence of adverse effects. Levetiracetam is a very widely prescribed drug, and we need more sophisticated predictive models to allow patients a much more satisfying and safer experience with anti-seizure medicines. And last, I'd like to thank people in my laboratory, particularly Amy Shakeshaft, whose work for her PhD thesis has been presented extensively in this talk. Thank you very much.
Deb, thank you for, as expected, a really very interesting talk. I found it fascinating that there is such a big stratification and sex-based difference. I do see people with JME in my general clinic. I wondered how these findings relate to genetic risk. Are there genetic factors that are underlying these differences that you see, these sex differences? Of course, if there are and you haven't published them yet, don't mention the genes, but that's the question really. Is there a genetic effect here?
Yeah, it's a great question, Ammar, and that's what we're working on now. I think there's a combination of a genetic and perhaps an epigenetic or environmental component to this sex difference. The reason that I've been talking about prognostic markers and stratification is because this is our approach to unraveling the complex disease genetics in epilepsy. And I think that the same things that turn out to be stratifiers are going to be really good targets for drug resistance in epilepsy, like absence seizures and so on. So to come right back to your question, in our GWAS of absence seizures, we have found a GWAS significant hit that is male specific. In other words, it only confers risk to men for absence seizures and doesn't have any effect on women. Regarding the stress sensitivity, I think this is really the most interesting part of the study, and this has just been published actually on this week in Scientific Reports. I think the interesting thing is that having stress-sensitive seizures makes women more likely to develop drug resistance, and I think there may be other things going on through an interaction with the HPA axis and with sex hormones, perhaps. We know from other studies that stress sensitivity can be increased in risk by previous stressful life events. So that's a direction that we're going to go in next, is to see whether what we're picking up in a cross-sectional study as stress sensitivity is actually an accumulation of lifetime exposures.
And so you've mentioned the presumably an X-linked gene, if it's only seen in men or predominantly seen in men. But if this condition is more common in women and there's a sex difference in risk that's really affecting women, where would you expect to see a genetic variant that's doing that, that's kind of women specific? Or are you saying it is really mediated by these hormonal changes?
Yeah, actually, it's not X-linked. It's an autosomal locus, but the risk allele is neutral in females. So we did a sex by SNP interaction analysis and found that the risk was only there in women. I think with larger sample, we will uncover more female specific loci, and also we'll have the opportunity to look for genetic or gene environment interactions. So some of these risk alleles or gene pathways may be involved in modulating the stress response, glucocorticoid receptors and so on. So I think there's a lot of interesting and exciting stuff beyond the ion channel to be discovered in these common complex epilepsies. And
do you see these kinds of changes in other epilepsies, or is it really something very specific to JME? Stress sensitivity is something that's seen across all epilepsy, but it's most pronounced in the
idiopathic generalized epilepsies, which are the adolescent onset ones, very common, about 40% of all epilepsy. But there is something about stress pathways or stress response pathways that I think are an important modulator of drug response.
Okay. Well, thank you very much, Deb, for a fascinating talk and a wonderful discussion. And I'd like to thank all of our speakers for keeping to time and for answering the questions so effectively and so well. Thank you. And I'd also like to thank Dr. Joanne Bell from the Brain team, who's helped put this session together. So thank you very much. Feel free to look at all of the posters and enjoy the site, and enjoy your lunch if that's the time zone you're in. Bye-bye.